Welcome back to Forest Talk, where we talk story about the plants and the animals and the connections that we have to our native ohia forests. My name is Kim Rogers. I'm an outreach specialist with Kauai Invasive Species Committee. My focus, as you all likely know, is ohia and protecting our native forest ecosystems from rapid ohia death and other forest threats. Remember, as always, I like to refresh our memories. A healthy ohia forest helps prevent erosion and ensure we have a healthy ocean reef in which to swim, surf, snorkel, and fish. It also helps to maintain a healthy coastal ecosystem for things like our native bees. A healthy ohia forest also helps ensure we have fresh water to drink. To help stop the potential spread of rod and other threats, before and after hiking, we ask that you use proper biosanitation protocols, which is basically simply brush as much mud off your footwear and your gear as possible. And the second step is to spray with isopropyl rubbing alcohol. If you would, admitting a couple more people here, during our time together, if you would follow good Zoom etiquette, keep yourself muted. If you have questions, please feel free to put those in the chat. We'll get to them at the end of our presentation. We are recording, as you may have seen, and I will send out the recording early next week, where we also have uploaded to our YouTube page all of the previous Forest Talk episodes that we've had. I'll put a link to that in the chat in a minute. So all of our previous 12 or 13, however many Forest Talk sessions have been recorded and are on the KISK YouTube page. I want you to make note of our next Forest Talk, so check your calendar. For Monday, July 6th, our guest speakers, there'll be two, will be Amanda Vernon and Tim Flynn from the National Tropical Botanical Garden. And they are going to share about the common mosses of the Hawaiian Islands. They actually have this cute little book they just published. If you want to register for that, if you haven't already, I'll put the link in the chat for that in a minute too. But first, let me introduce our guest speaker, Dr. Sheldon Plentovich. I first met Sheldon, some 10 or so years ago, I want to say, aboard a research vessel in Papahanaumokuakea Marine National Monument. Look, she's laughing. It was that summer we had like the 12 dozen tropical storms, and I think we had a tsunami, and I think we had hurricanes and all. It was a crazy, crazy several weeks. I think I was on that ship for an extra week or so. It was like 5 or 6
Was
weeks.
that when we got rescued off of Laysan or something?
It was. Yeah. Yeah. We dropped you off, and then like hours later, I think we were like, come back, come back. There was like an earthquake off of South America somewhere. And then we had the tiniest little run up of the tsunami, remember, the next day. Anyway, you were doing, I think, Nihoa Finch work, right?
It was Millerbird. It was
Millerbird.
Millerbirds that we had translocated. Yeah.
That's right. That's right. That's right. And I was there doing monk seal work. So that was like a lifetime ago, it feels like. But let me introduce appropriately Sheldon. Sheldon is a restoration ecologist with the U.S. Fish and Wildlife Service, where she holds the very cool position and title of Pacific Island Coastal Program Coordinator. And she works with partners to conduct collaborative ecosystem restoration. Today with Sheldon, she's done a lot of really cool projects, but today the cool project that she's going to share with us is about our Hawaii's native yellow-faced bees. And so with that, I'm going to turn it over to Sheldon. She's going to share screen. I'll let you know if that's coming up. And then I'll disappear and let Sheldon have everything.
Okay, let's see here. Hang on 1 second. There we
No
go.
problem.
No, it does not like what's happening.
Oh, of course. It worked 5 minutes ago.
It doesn't surprise me. There we go. Okay.
We'll figure it out. Okay.
Okay, let me know when it's in presenter mode.
It is in presenter mode.
Hooray, we did it. Okay. Thank you for that introduction. It's good to see you and to see, I recognize a lot of names on the call. And so aloha, everyone. Mahalo for this opportunity to talk about one of my favorite groups as Kim mentioned, I work with a lot of different species, but I have to say that Hawaiian yellow-faced bees are one of my favorites, and they are probably one of your favorites. And if they're not, it's just because you don't know enough about them yet. So hopefully this will help. So today I'm going to talk about, this talk is called Creating a Buzz, Protecting and Expanding Populations of Native Solitary Bees in the Hawaiian Islands. And I first started working on these bees when I was in graduate school at UH, getting my PhD, doing work on offshore islets. And I met Cynthia King. And that was like back in the Stone Ages, right? And Cynthia and I, years later, after we had graduated, she was working for the state and I was working at Fish and Wildlife. And we were like, hey, we need to do something about the bees. You know, they're just declining. Populations were disappearing. So we put our heads together in 2016. We got Jason Graham, who's now with Planet Bee Foundation, to work with us. And then as the project has evolved, now we're working more with Paul Crucial-Nicki and Janice Matsunaga. So those are members of the bee team. Okay, so I'm going to start with some general background on Hawaiian yellow-faced bees as a group, talk about their conservation needs, why they're so rare, and then focus in on one species, Hylaeus anthracinus, which is the one you can see in this picture, and talk specifically about three different things we've been working on. One is examining the effects of invasive ants on nesting Hylaeus anthracinus and looking at the relationship between bee populations and floral resources. And then finally, I'm going to talk about the results of our translocation efforts. And hopefully there'll be some times for questions at the end. But if you have a burning question during the talk, just interrupt me. I'm fine with that. Okay. Hawaii has a single group of native bees. So the vast majority of what you see out day to day are just introduced invasive bees. There is only a single group that are native, and they're all in the genus Hylaeus. There are about 63 known species in this genus. They're all endemic. They're only found in the Hawaiian Islands, and 7 species are listed as endangered, and that happened in 2016. They were once common and widespread, and this old school entomologist commented in the early 1900s that they are almost the most ubiquitous of any Hawaiian insects. But unfortunately, the majority of the 63 known species haven't been seen in decades. Many of them are very likely extinct, and the ones that we do know of typically survive in small, isolated populations. Not all of them, but most of them. And the reasons for that decline are primarily habitat loss. You know, as everyone on this call knows, we have limited native dominated habitat left in the Hawaiian Islands. You know, it's in small pockets and makes up about 31% of the landscape. Other reasons for this decline are loss of nesting and foraging sites through development and invasive species, which we'll talk more about later. And then, of course, when you lose habitat and these species disappear, that interrupts mutualistic relationships because, like many endemic species, Hylaeus evolved with elements of the flora to form really important mutualistic relationships that are unraveling or have unraveled. Other causes of decline include competition for floral resources and nest sites and other things as well. And that's by invasive ants and also introduced bees and wasps. And this picture here on the left is an introduced Hylaeus species. It's called Hylaeus strenuus. And although it looks quite cute in this picture, it is kind of pushing out Hylaeus anthracinus in some of our sites. Other causes of decline include predators. You know, invasive ants not only compete with Hylaeus, but they also depredate Hylaeus. And we'll talk more about that. Also, predators like vespid wasps and assassin bugs, which you can see in this picture, an assassin bug that has killed a male Hylaeus anthracinus, and then other introduced spiders and things like that. And then we're seeing that parasites and pathogens are more of a problem than we originally thought, and also pesticides. Okay, so obviously it's things are just kind of bleak for, a lot of our endemic species. But for insects especially, as someone who spent 10 years sorting through insect samples from lowland areas in the Hawaiian Islands, I can tell you that less than 5% of the samples I looked in were native species. And so it's a crisis, as we all know. So when Cynthia and I first got together on this back in 2016, our goal was to develop applied management tools to increase populations of yellow-faced bee species, which seemed impossible at the time. So we focused in on the one species that we were most familiar with and the one that we had pretty easy access to known populations. And so that was Hylaeus anthracinus. This is one of the species that was listed as endangered in 2016. You can see in this picture, in this illustration below, that the male has yellow markings on the face, the female has a solid black face. And this species is limited to coastal strand. And when I say limited, I'm not messing around. Like, it is a very narrow band of habitat that starts at high tide line and extends Mauka until there's development or a change to invasive dominated flora. So this picture is from James Campbell National Wildlife Refuge. That is a predator-proof fence on the Mauka side. And this is one of the few places where, as that coastline erodes away, it will be able to migrate inland, and we will still have habitat for the species here. But this is the exception, not the rule. Because they are in that area just above high tide line, they are especially vulnerable to storm surge and large wave events. And this threat, of course, is exacerbated by climate change. This is a picture from Turtle Bay Resort. This is a peninsula that's kind of right in front of the resort. And we were monitoring this small population of Hylaeus anthracinus at this site when a large wave event came in and extirpated the population right as we were monitoring it. We were there one day, and then back 4 days later, all the bees were extirpated. Okay, and so currently on Oahu, we have Hylaeus anthracinus is limited to the north coast of Oahu, Marine Corps Base, Hawaii, Ka'ivi coastline. We have a nice population there. And then there's a smaller population, like more localized population at Ka'ena Point. And so, our goal, which you already saw, was to develop these applied management tools to increase populations. And so to do that, we wanted to do three things. We wanted to identify limiting factors, find ways to augment habitat to increase the population and distribution. We don't just want to study its decline. We're really hoping to increase this population. And then finally, we wanted to establish additional populations of Hylaeus anthracinus through translocation. So we'll start with the first one, identifying limiting factors. One thing we were especially interested in is nesting success. And, we found nests when we were out in the field, but they, like at the Ka'ivi site, on the south shore of Oahu, they're nesting in coral rubble, which is odd. We hadn't found that before. We think that's probably what they used to nest in a lot, but because there's so many invasive ants now, they really can't do that. But there's low densities at Ka'ivi, so they are still in coral rubble there. And then, but generally throughout the rest of their range, they're nesting in hollow twigs. And you can see on the left is a nest that's been dissected out of a hollow twig. Don't worry, no bees were harmed in this. We reared these bees to adulthood, but you can see that it's really difficult to study natural nests. We know from the literature that they nest in dead hollow stems, holes in the ground, coral rubble. And we had anecdotal observations that nest sites were limited in some areas because the bees can't hollow out a stem themselves. They don't have, strong enough mouth parts for that. So they depend on other processes to hollow out stems. So there was some thought that maybe, they were nest site limited. So we were interested in that. And then, as we started monitoring these trying to monitor these natural nests, it was it was pretty much impossible to get much meaningful information from it. So we were interested in an artificial nest, which had been used, on the continent and elsewhere. And we know from a study that was done in 1982 that females would nest in substrate with pre-drilled holes. So we were like, let's see if we can make that work. So we made these nest blocks that you can see here, super high tech, right? Just 2x4 or 2x6, and then drilled some holes in it. But we put this removable plastic in it, and the holes were drilled based on measurements we had gathered from a lot of different natural nests. So we tried to give them exactly what they wanted. And the females readily used the tubes, which, resulted in wild dancing in the field and things when we saw that happening, because that was really cool, because then you got to watch these nests from the very, beginning of their construction all the way to the end. And I just thought this was the coolest thing. So here is a female Hylaeus anthracinus. She has 3 cells. What she'll do is she'll come in here, and Kim, I'm assuming you can see my pointer. Is that right?
Yes, it's super tiny, but yes, I can see it.
Okay. Okay. So she'll come in and she'll start with this cellophane-like membrane that she'll put at the back of her nest, and then she'll provision with pollen, and then she'll lay an egg, and then she'll seal that off with another cellophane-like membrane, and she'll provision with pollen, and she'll keep doing that. And so you can see here, there's probably 3 eggs have been laid. This nest is still under construction, because once she's done, she's out of there. And we had numbers of cells anywhere between 1 to 8 in these nests that we were monitoring. And it was really cool. You could watch things develop. The egg hatches, it's a larvae at that stage. And then the larvae pupates. And then this is kind of a middle stage pupa. It's got the dark eyes. They're all white, then they get dark eyes, and eventually they get dark bands on their abdomen, and then they'll turn black. And this is kind of an opaque plastic, but these are all healthy pupa that are just finishing up. And you can see the white pupa are still in these front cells. So from time to nest initiation to these animals leaving the nest as adults averaged about 37 days and ranged from 29 to 52 days. So the nest blocks gave us a tool to track nest success. And so we were then able to ask some questions that could help us identify limiting factors. So we were really interested in the impacts of invasive ants on nesting Hylaeus. We know that Hylaeus populations are suppressed in areas with invaded ants. We know that invasive ants compete with Hylaeus for floral resources. We know that ants depredate Hylaeus, but we just, had no real understanding of how they affect nesting success. So, Cynthia and I got together with Jason, who was making the nest blocks, and we hypothesized that if we could exclude ants from some blocks and not others, we could answer some questions. So we hypothesized that nest blocks that excluded ants would have increased nesting success and produced more adults compared to nests in blocks that could be accessed by ants. And so we used a paired experimental design where we put a control and a treatment block out within a meter or 2 of each other. And the treatment block was treated with tanglefoot so that ants could not access it. And then the control block was just left without tanglefoot, and ants could easily access it. The blocks were placed in pairs at 22 points across 3 sites. And the 3 sites were James Campbell, Turtle Bay, and Kaiwi. And so that's James Campbell, that's Kaiwi, and that's Turtle Bay. And then remember from the previous slide, Turtle Bay got washed out. So none of the data were used in the results I'm about to show you. Okay, So the sticky barrier was used to prevent ants on the treatment block. You can kind of see it right here. It was just a little gooey barrier right on the rope. And then this was the control block. It was left available to ants. And the blocks were monitored twice a week for a year between December in 2016, December 2017. And then when we went, we recorded species, number of cells, presence, absence of ants and adult bees, and then notes on development. And, you know, this was a while ago. And it's the first time people were really looking in these nests. And at first we thought it would be really straightforward and easy to tell what was going on since we were checking them twice a week, but it ended up being not like that. And so we resorted to drawing what we saw, which is what you can see here. And that was really a good way for us to track nests. And it was cool, you know, it's kind of a wonky nest. It's got this weird first cell back here. But here you can see this was when it was brand new. This is, you know, after the pupa, after it's pupated. And here is a pupa with those black lines I was talking about. This one's a little older. This one's stuck in the pollen, which happens sometimes. And then here on the bottom, you can see this animal is probably ready to leave, and this one's getting close. So it's really cool to be able to see the nest progression over time, except for we also, unfortunately, saw ants pillaging a lot of the nest. So in all, we observed 920 nests across the 3 sites. 70% of the nests in the control blocks were invaded by ants. And nests in the treated blocks, which in theory ants did not have access to, were way more likely to produce at least one adult than nests in treated blocks. I do have to say that, not surprisingly, ants found a way sometimes. And if it was windy or a stem went across the block, sometimes ants did breach the treated blocks, but it was pretty rare, thank God. Okay, and then in the untreated blocks, ants were the most common cause of failure. Okay, so don't worry too much about all this data. I just kind of want you to look at the general, the similar results in both of these graphs, or both of these graphs. The difference, they show the difference in the number of successful nests on the left and adults on the right, produced in treatment and control blocks by site. So remember, it's a paired design. And so numbers above 0 indicate points where there were more successful nests on this side on the left, or more adults in this on the right. And so you can see across all 22 of the locations where we put out both blocks, you can see a big difference between the treatment and the control. Okay. Glaber ants, or Ocatellus glaber, they're the little black ants that are often in the vegetation. They do bite or sting you if you've spent some time with them. They'll do that when provoked. But they were the only ant species we observed depredating Hylaeus nest. And this surprised us because at the 3 sites, Pheidole megacephala, or big-headed ants, were the most common species. They were abundant, pretty dense, but on the ground. And it seemed like the 2 species of ants were partitioning the habitat, where big-headed ants were primarily foraging, nesting on the ground, which is normal. And then Ocatellus glaber were nesting and foraging in the vegetation. So we do know from other sites that big-headed ants have been found, like depredating Hylaeus, but we didn't see that at our sites. And interestingly, or sadly, while we were doing this study, yellow crazy ants were invading James Campbell from the east, and they ended up invading 3 of our points at that location. And so there were no nests, and virtually no adults were observed at the 3 points that were invaded by yellow crazy ants. So, you know, we don't have time to talk about all the data we collected, but for one of the projects, we were doing adult counts. And so at these sites where yellow crazy ants were, we weren't finding any adults either. So they just cannot coexist. And here's, even though like we didn't see, there were no nests that we saw that were depredated by yellow crazy ants, but yellow crazy ants were just, they were climbing all over the nest blocks. And so I think even if a bee had tried to nest in these blocks, the yellow crazy ants would have just removed, any evidence of that pretty quickly. Okay, so in summary, we did find support for our hypothesis that nest in blocks that excluded ants, they did have increased nesting success and produced more adults than nest in blocks that could be accessed by ants. Artificial nest sites were readily used, potentially indicating a lack of available nest sites. And then ants were the main cause of nest failure, followed by lack of development, nest takeover by this vespid wasp, Pachydineris, and then pathogens. And then, our limited data, we only have those 3 points, but they support previous claims that Hylaeus cannot survive in areas invaded by yellow crazy ants. And that is what has played out since then as the ants have continued to march westward at James Campbell. And if you're really interested in this, this work went into Neobiota in 2021. Okay, so then from there, we're like, okay, we got a little information. Now what are we gonna do? And so we improved the nest tube design. So because we found that those tubes, when water got in there, it just wasn't able to evaporate. And that was not good for the nest. So we ended up using perforated designs. We tried a bunch of different things. We switched to the bee condos, which you can see here. And so there's a cuff here at the bottom, and tanglefoot was kind of hidden under that cuff, just so the bees couldn't interact with the tanglefoot, just the ants. We improved ventilation, as I mentioned, in the tubes. We made them slightly smaller so that invasive wasps couldn't use the tubes, and that worked amazingly. And so we continue to collect data using those bee condos. The main thing we were interested in is if there was limited nest sites, could augmentation of nesting habitat with our condos increase adult population? And unfortunately now, we've been doing that for about 5 years, and our preliminary data do not show an increase, unfortunately. And the next thing we were interested in is looking at the relationship between floral abundance and bee abundance. Is the number of flowers a limiting factor for the bees or not? And then we've also been examining the pollen used in the nest, trying to determine if the pollen is it all alpaca? Is it a mix? Do the bees like some things more than others? And in general, I'm not going to present all the data on that, but in general, what we find is that the bees use what's there, but they will go out of their way to get a coolie, which is Sesuvium portulacastrum, or a cocoa, Euphorbia degneri. They freaking love those two, and they will, go to the ends of the earth to find those two plants. So we're trying to give them more of those in our sites. And then the last thing we wanted to do was identify coastal field sites that could be suitable for bee habitat enhancement and or reintroduction. And so for the rest of the talk, I'll talk briefly about 2 of these. But if you have questions about any of them, reach out. Okay, so let's talk about relationship between floral abundance and bee abundance. So to do this, we established 10 meter by 10 meter plots. We surveyed those plots quarterly, and we estimated flower density for each flowering species within the plot. We also estimated veg cover and volume. And then at each month, we did timed counts of Hylaeus. And here's just a piece of this data. So this is time on the x-axis, mean bee count on the y-axis, mean total flowers on the z-axis. And this is only from James Campbell. And this is from the 2018-2021 timeframe, but it's what we find now too. So the red are the flowers, the blue are the bees. And you can see that as flower number increases, there's a lag time, but then the bees increase afterwards. And then conversely, as flower numbers decrease, the bee numbers decrease as well. And so you can imagine that these low points here are limiting bee numbers. And as you know, I'm sure most people on this call know, our coastal ecosystems are depauperate in terms of biodiversity or plant biodiversity compared to how it used to be. And so one way that we can support Hylaeus and other coastal species is just by reestablishing some of that biodiversity, and maybe we won't have these big dips in flowers, you know, because right now this is mostly driven, flower number is mostly driven by naupaka and beach heliotrope, which obviously beach heliotrope isn't native. So just something to think about. And so understanding that relationship led to, you know, other questions. And here, this graph is a little messy, but you're going to see it twice, unfortunately. And it shows the relationship between mean annual bee abundances, which is here on the y-axis, and mean annual flowers per plot on the x-axis. And all I want you to see from this one are the three sites. This is Marine Corps Base in the green. This is James Campbell in the red. And then this is the Kaiwi site in the blue. And so you can see more flowers, more bees, basically. Relative abundances of Hylaeus were correlated with flower abundances on an annual time scale. And the mean number of flowers per plot was a significant predictor of Hylaeus numbers. So that means when we go to a place where we're wondering if there's Hylaeus there, or we maybe want to restore it so that we can translocate Hylaeus, we can use mean number of flowers to give us a lot of information about how many Hylaeus it could support or whether Hylaeus are there or not. So more flowers equals more bees. The data show a strong influence in floral resources on Hylaeus anthracinus abundance across spatial, seasonal, and annual scales. A diverse plant community would provide consistent forage across seasons. So that's kind of our goal moving forward with restoration. And these data are relevant for identification of suitable sites for bee reintroduction, and they provide a target for plant restoration activities. Okay, so now moving on to the last thing. This is, we actually translocated Hylaeus several years ago. And it was interesting. You know, I've translocated birds before, but never bugs. So this was fun. And we do translocation to create new populations, which will buffer the species against extinction, and to reestablish those ecological functions that are lost when the species is extirpated from areas. In this picture, this is a female anthracinus, and she's on this Euphorbia degneri flower, which has these crazy flower morphology. Okay, so for the translocation, we looked at a lot more than 10 sites initially, but we kind of reined it into 10 areas on Oahu. We scouted the sites. We evaluated them based on plant diversity, how many flowers were there, which ant species were present, and if there was natural nesting substrates. And we ended up selecting 2 sites. So the yellow dots are known Hylaeus locations, and the 2 sites we selected were Kaleloa, the Kaleloa unit of the Pearl Harbor Refuge down here on the South Shore. And then this site at Kaena State Park that it had a nice diversity of plants, but you know, if you've ever been to Kaena State Park, not the natural area reserve, you know that it has been, it has suffered from decades of degradation from off-road vehicles and trampling and all sorts of problems. So I was kind of surprised when Paul came at me with this site. I was like a little unsure of it, but we're about to find out how it worked out. Okay, so to actually do the translocation, we chose to move developing nests from our bee condos rather than adult bees. Because the adult bees, you know, we don't know exactly how long they live, but it's not that long, you know, probably no more than a month. So we collected a total of 316 nests that contained 769 brood cells from James Campbell and Marine Corps Base, and we moved them in July and August. This is our team. And so each tube was removed from its original nest block, you know, at Marine Corps Base or James Campbell, and then placed in a paper envelope, transported in this really high-tech cooler. You can see the high-tech coolers you can see here to the release site. And we had put the bee condos up at the release sites. And so we just slid the tubes back in the holes of the bee condos we had put up at our release site. And, you know, the whole translocation took more than a couple of hours, you know, each time we did it. So each translocation site, Kalaeloa and Ka'ena State Park, they had 10 bee condos set up, and then we had already established the monitoring plots we were going to use. And we looked at floral abundance, ant activity, adult bee abundance at the translocation sites, you know, once the bees were there and just before the bees were there. And then the nest blocks were monitored weekly for 1 year after translocation. And then they were, after 1 year, you know, we had a site where the bees stayed there, and we just kept monitoring that site, and we still monitor it to this day. Okay, here's the results. The translocations went surprisingly smoothly. The adults emerged at their new location and constructed nests there at the new location within a week. So that was amazing. We had nesting persist at the crappy site at Ka'ena State Park at low levels, and they're still there to this day. But they ceased, they disappeared at Kalaeloa after about 20 weeks. Over the first year, we had 122 nests that were constructed at Ka'ena compared to only 42 at Kalaeloa. And so here you can see time, this is weeks across the x-axis, and then adults alive per week on the y-axis. So Ka'ena is in blue and Kalaeloa is in red. And so you can see initially we had similar numbers emerge, and then Kalaeloa numbers just crashed after about 20 weeks. And, you know, Ka'ena numbers crashed too, but then they sort of stayed at this low density all the way until now. So here you can see, again, time on the x-axis, adults emerging per week on the y-axis up top, and then mean bee count. So that's the count of adult bees out in the wild. And so many more adults emerged at Ka'ena. You can see the blue here, you know, continuing all the way to the end of the timeline, whereas at Kalaeloa in red, they just kind of disappeared. And so despite like these similar initial emergence rates from the translocated nest, adult bee counts were much lower at Kalaeloa than Ka'ena. So you can see this is the Ka'ena with the dark circles compared to Kalaeloa with the open circles here on the bottom. So we were kind of confused by this because the Kalaeloa site, you know, it had like, it was dominated by Nio, Myoporium stellatum. And we know the bees like that plant. We haven't ever seen nesting in it. But that site, there were way more floral resources at Kalaeloa. So here's the open circles. You can see this is just number of flowers on the y-axis, and again, time on the x-axis. So you can see that just crap load more flowers at Kalaeloa. So we don't think that forage was the limiting factor that caused the failure at Kalaeloa. So we're like, well, maybe it was ants, you know? And so we looked at ant density, and you can see that this is two different ways of sampling ants, beating and baiting. You know, beating the vegetation gently, not destroying it, but beating it just enough for the ants to fall off, and then baiting using like bait cards. And, you know, there are more ants at Kalaeloa, but we didn't think that it was a big enough difference to contribute to the major differential success we observed. So we were trying to figure out, other possible reasons for failure. And we did have a single brood that was parasitized by a bomb-billed fly. And so brood parasitism is just when it's either a fly or a wasp, and they'll lay an egg in a nest, and their larvae will eat, the nest of Hylaeus, basically. And so we had one single brood cell parasitized at Ka'ena, but at Kalaeloa, we had 8 independent colonizations of nest blocks by 2 different wasp parasitoids, basically. And this impacted 10 nests, which represented about 24% of all the nests at Kalaeloa. So if you take parasitism out of the calculations, the nesting rate at Kalaeloa would increase from 44% to 56%, which is, way more similar to Ka'ena. So we think that this is a likely contributing factor and, a limiting factor for the species at some sites. So why did Kalaeloa fail? We think it was the high levels of parasitism. It could have had something to do with lack of nesting substrate. Like I said, we hadn't seen nesting in Nio, but we're not 100% sure about that. Whereas meanwhile, back at the crappy site at Ka'ena State Park, we've seen this is nest number here. We've seen continued nesting and then continue adult bee counts through the present. And so recall this graph that I showed you before with mean number of flowers on the X, mean bee count on the Y. And so here we've added Ka'ena State Park. So it's got hardly any flowers. And so, of course, that makes sense that it also has a really low density of bees. All right, and this is a satellite image of that site. You can see there's so much bare ground exposed. People are trampling it and still, riding dirt bikes through our restoration site. And so to this day, we have low bee counts at Ka'ena. That's consistent with the low floral abundance at the site. But, we put in a lot of energy doing restoration here. We've tried to put symbolic fencing up around the entire site to keep people out. But I'll sit there and watch, and people will just walk right over the rope and just walk in and trample on our plants. Like, this site is taking years off my life. But before I die, we're going to get the site restored. Okay, so in conclusion, the establishment of new yellow-faced bee populations through translocation of developing nests was pretty straightforward and effective. The presence of parasitoids and maybe a shortage of nesting substrate at Kalaeloa are possible explanations for failure at that site. And even with limited floral resources at Ka'ena State Park, the bees have persisted, and they're still there, and they're getting more and more plants, so their numbers are increasing. Plant restoration informed by foraging observation and nest pollen analysis could improve Ka'ena State Park, and what we're learning there could make additional areas suitable for similar translocations in the future. Okay, last few slides. So recall that Cynthia and I's original goal was to develop applied management tools to increase populations of yellow-faced bees. So let's see how we're doing. So we wanted to identify limiting factors, see if we could augment habitat in a way that increases population, and then see if translocation to restored sites could be used effectively. So for limiting factors, what do we know now? We know invasive ants are limiting factors for adults and nesting Hylaeus, especially yellow crazy ants. We know that floral abundance and diversity are also limiting factors. Remember, you need high numbers of flowers to have lots of bees. Nest parasitism is a problem. Invasive bees and wasps compete for floral resources and for nest sites. And then habitat with suitable nest sites is likely a limiting factor at some sites, but we're still not clear on where and why. Okay, and then the second thing, can we augment habitat in a way that increases the population? What do we know now? we know that we can restore plant diversity and that they especially like a cocoa and a kuli. So we're actively adding that to the sites where we have bees. We can control invasive ants, which is a challenge, but is possible. We've done it before and we're doing it at some sites. And we can expand restoration areas inland, like that picture I showed you at James Campbell, so that the coastal strand has a place to migrate to as sea level rises and our beaches erode further. And then could translocation to restored sites be used effectively? Yes, I think we showed that we can translocate developing nests. It's pretty straightforward and effective. It was actually pretty easy, given these other translocations I've done with birds. Adult bees are persisting at that one site at Ka'ena State Park. If you're interested in where it is, it's right. If you park at the main parking area there at Ka'ena, it's just kind of right in there from the parking lot. It's like 200 meters in. And then we can now better evaluate potential sites to ensure that they include adequate nesting and floral resources for future translocations. So we made some progress. It's important to point out that this is one of the approximately 63 species. We've done a lot of work on this species and we do know a lot now, but there's another coastal species, Hylaeus longiceps, which is what you see here. And I think this species probably thinks we have a lot more work to do for this one and for all the rest of the 63 species. So that's it.
Thank you. I am going to, oh good, we're getting some people using their reaction buttons, sharing the claps and the hearts and the thank yous. And why don't we maybe stop sharing your screens? We can everyone see each other. And this would be
hang on 1 second.
Yeah. Perfect.
Tell me when I'm stuck. Did I stop sharing?
You did it. You're great. And we have a question in the chat, and then I have, a bunch of them. Because the whole well, and what's funny is I write a question down, and then your presentation goes on. I'm like, okay, she answered that one. Okay, answered that one. But that was absolutely fabulous. I really, really cannot thank you enough for sharing, because these cute little bees, oh my God, and they are so cute, right? We just do not know enough about them. And I feel like there's a whole lot more we could be doing. And so with that, thank you. So
my pleasure.
Yes.
Everyone should know more about yellow-faced bees.
I know. They're so cute. And I love that how successful you were with a translocation and then creating the nesting tubes and
that was so fun.
Right?
Yeah. It's been like super fun as a biologist, works on all sorts of other things. It's just been super fun and fulfilling to do it. And what was key in getting people to care about it was getting the pictures.
Oh, that was the other note I made. Your photographs are amazing, right?
So that was Jason and I in a picture war trying to see who could get the best photographs. And once that happened, it was wild how many people became more interested in them because they could see them and they look almost relatable,
Yeah,
like
they're cute.
A little male bee with a beard. And
they're charismatic, right? It's that whole, oh God, Diane, great. Did you see Diane's?
Yeah.
Bee going up. Fabulous. Okay, that is one thing I want to point out. I don't think you mentioned it. If you did, my apologies. Our native bees are not colonizers, correct? And they do not make honey.
They do not make honey. They are solitary nesters. We do see, like, when there's good habitat, there's often a lot of them. And so people see them like swarm. Like at Ka'iwi, there's this area of coral rubble where you can kind of see them swarming almost. And so it confuses people. They think they're colonial nesters, but they're not. They're solitary nesters. They don't sting. However, I will tell you on the Hoa, that bee, that Hylaeus perkinsianus, has stung me multiple times. However, I have asked for it every time. It gets like under like my neck scarf or something, and then it'll give me a sting and let me know, hey, quit squashing me.
Yeah,
but
I'm here.
Our Hylaeus, Yeah, but
Yeah.
our main Hawaiian island, Hylaeus, I have never been stung by, and I have been in situations where I deserve it.
Well, I think those are really good points to make up front because we as a people, the bees that we're used to, we so associate bees with honey and colonies, so just wanted to clear that out. And we're getting a bunch of great questions in the chat, but
Okay.
I'm going to start with one of mine first. And I know we were focusing on coastal bees today, and there is, as I always talk about, that relationship with a healthy ohia forest helps ensure a healthy coastal reef and ecosystem and all of that. So totally connected. But I am curious, are there bees, yellow-faced native bees, found at our higher elevations, or how high of elevation do you know that they might be found? And you're nodding.
They're all the way up in Haleakala, like Paul Crucial Nikki they pollinate silver sword.
Oh,
They, they're
seriously?
all the way from the mountains, to the ocean.
Oh my
Um,
gosh.
and so it's hard, right? Because our upland areas have some intact forest and intact ecosystems, but they're harder to access, and we just don't know a lot about the bees up there.
Okay, well
They've been overlooked.
that is inspiring me to go back up to Koke'e, Iliau
Ooh.
Trail, where it's at the very end of the season, but our Iliau are blooming. And they're always,
Ooh.
I mean, I always see all of the bees around them, and I honestly just assume they're non-native. So I'm gonna
Yeah.
go back.
So in Koke'e, I had bee experiences on some of the lobelioids,
Yeah.
and that are definitely Hylaeus up there, but they are small. So one thing that underwhelms people, especially Maisie Hirono, when we took her out to Ka'ivi, her eyesight isn't as good as it used to be, like all of us.
Right.
And she was really bummed about how little the bees are, because they are like, they're not even an inch long, right? So people can easily confuse them for flies.
Okay, so
So
that
just
was a question in our chat.
be
So
aware of that. Yeah.
under an inch, but honestly, that's still a good size. So yeah.
Totally.
All right,
Yeah.
so here we have a question from Joanne, who I think was from North Carolina. I'm curious, are dragonflies and skimmers prevalent in these areas, and are they a natural predator?
Oh, dragonflies. Yeah. So we see dragonflies. We see dragonflies at all the sites where we have Hylaeus. We have native dragonflies. And I have never once seen any kind of predation event by a dragonfly. However, like you saw that picture of the Reduvia, the assassin bug. Like,
Yeah.
I've seen some crazy stuff happen while I'm in the bushes looking at Hylaeus, but I haven't seen dragonflies do anything.
Okay,
Yeah,
well,
or skimmers. Yeah.
okay, good to know. And then Dana is asking, are there still plans to make Ka'ivi into a park? By using artificial bee condos over time, could you possibly be directing selection towards dependency on human intervention in nesting? And regarding translocation, different plant populations may be the same plant species, but different chemical composition in pollen, so more or less attractive to translocated bees. Take that.
Okay, let's go one. Let's go one at a time. You're going to have to help me
Yeah, no
remember
problem.
all those three questions. So the first one is it a park? So Ka'ivi is in conservation status. It is conservation land now. It will not be developed. I myself would love to see, like, I think of Kilauea Point when I look at Ka'ivi coastline with the Makapu'u Lighthouse and all of
Right.
that. And I would really love to see in the future a predator fence and that entire coastline free of mammalian predators and just full of all the things that should be there, like more Hylaeus, more plants, nesting seabirds, all that stuff.
Yeah.
So we're getting there, but it's still way down the road.
Okay.
So question number one. But question number two is about, if you're using the bee condos, are you driving towards independence?
Or
We're
deriving towards dependency.
Towards dependence. Sorry.
Yeah.
We are using them in such a small area, and we're actually stopped using them when we realize we've analyzed the data and we see preliminarily that it wasn't translating into increases in adult population, even though bee condos were being used a lot. But they do seem to be able to find, this species seems to be able to find nesting habitat. And even on our control blocks that ants had access to, the success rate was about 20 to 30% of the nest were still successful. And that seems to be enough to sustain those populations. But overall, the areas where the bees are present is just a tiny little percentage of what it used to be. So, and where we were using the bee condos was just a tiny percentage of that. So we're not really worried about, we're not using them at a landscape scale, and now we're not using them at all. So no.
Okay.
And then what was the last question?
It was regarding translocation. Different plant populations may be the same plant species, but different chemical composition and pollen, more or less attractive to translocated bees. It's not written as a question per se, but I'm thinking that she's pointing out the difference in the chemical composition of pollen in the same plant, but in different locations. I think I understood that correctly.
Yeah. So we do see, like, for example, naupaka and beech heliotrope are the dominant pollens used in the nest. And even in the translocated population, that is what we saw. And in general, those are the things that are most prevalent at the site. Although at the translocation site, there isn't tree heliotrope. It was mostly naupaka that was being used. But we're not really looking, at any kind of chemical analysis of the pollen or anything like that. We're just like, nothing is known about the bees, so we're just starting at square one, right? Like, what species is being used?
Yeah.
And they tend to use what's there, except for a cocoa and a kuli, and they will seek that out. They'll fly a long way to get that.
Okay, so I have a cocoa that loves my yard where I live in on Kauai. I should be out there looking, right?
Yeah, look. I mean, if your yard is in a native forest, look. But
I mean, I have a bunch of natives, but it's not a native forest. Okay, I'll look though.
Yeah.
Anne asks, are there native plants that are dependent on this species for pollination?
Great question, Anne. And so Dave Hopper, Dr. Sheila Conant was my PhD advisor, and Dave was a student before me and looked at Ojai, or Sesbania tomentosa, and found that Hylaeus was the only effective pollinator of Sesbania at that time. That was 95. And since then, we've had two other little bee species have been introduced over here, Seratina smaragdula, and then we also have this Hylaeus strenuus, one that I showed you the picture of. And those bees can get into the business parts of Sesbania and pollinate. But prior to the introduction of those species, we know that Sesbania could only be pollinated by Hylaeus. That's all I know offhand, but undoubtedly there are other species that depend on Hylaeus for introduction, for pollination.
Right. And Dana just made the comment, absolutely delicious, which we're all going to have to remember and use.
I love y'all.
We talked about size, so we answered that question, Sharon. And Ku'ule shared that the first time she saw them was on Mauna Loa on Ohia.
That's so cool.
Off in the blossoms, which I love. Oftentimes that we find here, at least in Kokee and the mountains where we've got hiking trails, the ohia are so tall that when they're blooming, they're way, 60, 80, 100 feet up. And so how do you know, right? But again, I'm going to go back to Iliau Trail because they are younger and they're blooming at a lower elevation.
And Lori Lack did a study looking at ohia and Hylaeus that you can go back and look in her last name's Lack.
Okay.
And she was one of the first people to find that if ants were present on the ohia blossoms, the bees were less likely to come and take nectar or pollen. So you can look up and see where I feel like her study was probably on Kauai, but you can double check.
Okay, I'll look for that. And let's see. So that reminds me, seasonality. They don't live very long,
yeah.
but is there a seasonality?
Yeah, seasonality is huge. So if you go out, like if you guys came to visit me and I wanted to take you to see bees, if we went in August, I could show you bees swarming basically. And but if you came in January, we might not see anything.
Mm,
So, and it that can be different from year to year as well. So it's definitely, they just kind of go with the flow of what those flowers are doing. And if there's lots of flowers, we're going to see lots of bees. And then, so after the wet season, when the plants are responding to the wet season, you will see a lot of bees. And then, during the dry season, when the ant plants are kind of crispy and not flowering, you're not going to see much. So we're trying to find plants to get into the habitat that are still producing flowers during the dry season so that we can support the populations.
that makes perfect sense. Would that be a cocoa and a kuli kuli too?
Yeah, those are both plants that we have. They do have flowers year round, but they also, a kuli kuli has a lot of flowers during that dry season, but a cocoa is kind of finishing up. So it's
Okay.
but we're looking, and we're really focused on diversifying our coastal ecosystems here and just trying to get it back to a semblance of what it was, before it became so degraded.
Sure, sure. No, this is all like we're learning so much. And there's what I love about it too, because, when I work with students, there's so much more to learn. So, I tell young people, like, hey, you want to work here and stay home? Like, here's like this huge, wide open opportunity
Yeah.
for work, science and research that needs to be done that we could all learn. So,
Every question we answer about these bees, we have 20 other questions. It's
exactly.
just the more you know, the dumber you are, right? Like,
Right, right. Uh, So I'm wondering if you or anybody who's in the room has any mo'olelo or any cultural stories or connections they've come across with our bees, mele, oli, what have you? Sheldon, have you come across any?
that's a great question. And so we put some effort into this, and we couldn't even find a Hawaiian name for the bee. Um, and so yeah, we haven't been able to find anything. That's not to say it's not out there. We just haven't had time to put a lot of effort into that. But if anyone knows of anything like that, I'd love to hear it.
Yeah, me too. I think that would be great. So if anybody, is not able or wants to go off mute, feel free to speak up. Okay. Meanwhile, I know we should probably wrap up, but I have two last things and thoughts. Oh,
Okay.
wait, a question. How far do they forage and nest? Great
Ooh.
question. Thinking about the Kalailoa site, maybe they moved to a nearby site with a kule kule and a cocoa. Hmm.
Those are great questions. So I'll do the Kaliloa question first. So if you've been out to Kaliloa, it's don't ever tell anyone at Fish and Wildlife I've said this, but it's a very sad refuge. It's bound by development on either side.
Oh.
It's got garbage and fishing line all on the shoreline there, but there is a kuli kuli. We did put them kind of outside our fence there. So there would really be no place for them to go there, unfortunately. I was going to point out on the map I showed you that had the yellow dots for all the places we know they occur. The whole South Shore is just not yellow.
Uh,
So unfortunately, Kalailoa is a nice little spot in there, but it's not surrounded by anything but development. Now, her question about how far do they forage, that's something that Paul and I argue about all the time. We did a mark recapture where we captured the bees and we put a little touch of color on their thorax, and then we recaptured. And I think in general, they're staying within 5 to 10 meters.
oh,
However, we did find evidence that they traveled up to about 20 meters to get a plant they wanted. And we had this other weird situation where they had Plukia carolinensis pollen in a nest. And I could not find Plukia anywhere around the site. And Paul finally found it, it was like 30 or so meters away. And so there's that. It's just one example, but we don't think that that's common that they go that far. We think they just hang out in their spot.
okay, fabulous. All right. So, um, I have more questions than we have time for, so I'm going to limit myself.
That's okay.
Is the
Okay.
challenge finding, like I would imagine if they're nesting in little, hollowed stems and they're less than an inch, in size or in coral rubble, I would imagine finding their nest sites is challenging. And I'm wondering if dogs, conservation dogs, could help. What do you think?
Yeah. So I'm a bird nest finder. Since I was little, I've been into that. And so when given the opportunity to find the nest of Hylaeus, I absolutely loved it and thought it was pretty easy. You just got to sit
Oh,
there, and instead of walking around in the forest, you get to sit at one plant and be like, oh, there's one, there's one, there's one. So it is pretty easy to find them.
oh,
They just disappear. It's usually like a naupaka twig that's broken off and it's been hollowed out, and they'll just disappear into it. But the thing is, you can't see what's going on in there. Are they resting in there? Are they
mm,
nesting? What are they doing? You can't tell unless you dissect it, which is so destructive, right?
right,
So
right.
that's why we were stoked to get them nesting in the artificial nest so we could actually see what was going on.
Yeah, that was a stroke
Yeah.
of genius. And thank you, Kule. You are actually asking the last question that I was going to ask. So I'm gonna use Kule's wording. So if we plant a bunch of natives at home, is it possible to have Hylas in our backyards? And if so, is there anything we can do to help them and their populations?
So I think the way to help Hylaeus right now is to support ecosystem restoration, support recovery of native assemblages of plants, support predator removal, ant eradications,
Right.
things like that. And, if you happen to live like adjacent to, say, Kahuku Golf Course or something, you probably could get Hylaeus by planting plants. But I think if you're, living disjunct from naturally occurring Hylaeus populations, the only way you could get them is through translocation, which, wouldn't be permitted at this point. But that doesn't mean in the future there couldn't be ways to do that, you know, and recall at the beginning of this talk, I said Perkins, the old school entomologist in the early
Yeah.
1900s, it was the most ubiquitous of insects. Like if you read his accounts, like they were just everywhere and just swarming. So, I mean, it's a dream to get back to a situation like that, where we could have native species in our lowlands that were common, but right now, like you would have to like seek out Hylaeus to see them, as opposed to just attracting them to your yard, which could be, an aspirational goal we could all have.
Well, and it sounds to me like as well, like if we had corridors, right? And
Yeah,
if
totally.
we're in a neighborhood and we get like all of our neighbors and maybe we're close to a more intact ecosystem that hasn't been developed, does not have concrete over everywhere, maybe we can create like corridors, which that would be cool.
100%.
I want to
In
live
our,
next to people who do that.
I know, me, right? All of us. Yeah,
Right,
we just
right.
gotta reverse the tide that's happening. We're just like, we're in an extinction crisis. We're losing so many things. Like we just all have to work together and find ways to like push back on this and make things go the other way.
Okay, well, I'm committed to doing that. And I bet the
Same.
people who are here today are willing to join us in that effort, Sheldon. And one last time, I want to say thank you so very much. And the rest of us on the call, if you want to give another round of wonderful reaction buttons,
My
that would.
pleasure. Like I really love sharing about the bees, and just having people interested just like makes my black heart melt. So I really appreciate it.
Well, we can tell your enthusiasm is there, and it has not lessened over the many years that I've known you. So you keep adding cool projects to your, resume, so to speak. So thank you. Thank you so much for all you do. And we'll be in
Me
touch.
too.
We'll stay in touch.
Okay.
For the rest of
Aloha,
you.
everyone.
Yep, aloha everybody. I'll be
Have a great
editing
weekend.
the video and sending it out probably Monday. Hi, Colleen. Aloha. Have a great weekend.